Hello and welcome to another episode of Hema Now, our hematology podcast series. I'm your host, Dr. Jonathan Sackier, and in each episode I have the pleasure of speaking with leading voices in the field of hematology. Today, I'm thrilled to be joined by Dr. Paolo Gallipoli. Dr. Gallipoli is a clinical reader at the Centre for Haemato-Oncology at Barts Cancer Institute, Queen Mary University of London, and an honorary consultant hematologist at St. Bartholomew's Hospital. He completed his medical training in Naples before pursuing his PhD in experimental hematology in the United Kingdom. He's held prestigious fellowships from the Wellcome Trust and Cancer Research UK, and his research focuses on the biology and treatment resistance of myeloid leukemias, especially acute myeloid leukemia. Stay with us as we discuss his career journey, delve into the metabolic intricacies of leukemia biology, and hear about the cutting-edge research that could shape the future of acute myeloid leukemia treatment. It's a pleasure to welcome Dr. Paolo Gallipoli to the podcast. So, Dr. Paolo Gallipoli, welcome to the podcast.
Hi, good morning. Thanks for having me here today.
Excellent. So we like to start with some nice, gentle, quick-fire questions and just to get to know you a wee bit better. If you could have dinner with any scientist or medical pioneer, past or present, who would that be and why?
Well, that's a difficult question, obviously, because there's so many to choose from. I had a thought about this, and I think that the most likely, probably for a variety of reasons, is Donald Thomas. So Donald Thomas was the pioneer, together with others, but he's considered the pioneer of bone marrow transplantation, which, as you know, is now a routine procedure we carry out in many centers in the world, including where I work, and through which we actually saved the lives of many people. But obviously, when he started to work on that, it was considered to be unfeasible, impossible to do. And he had to face a lot of challenges. And if you read his life history, it's quite fascinating, both from a personal point of view, how he managed to become a doctor and then go into research, despite a lot of financial difficulties at the start of his life. And then afterwards, facing all the skepticism around his ideas and the resilience through which he accepted failures, because he had a lot of failures, and still managed to get this through to the point that now it's obviously become routine procedure. And he won a Nobel Prize eventually for it in 1990. Yeah. It's quite, I think, useful for a physician scientist or for a doctor, probably for any person working, trying to push the boundaries forward. That often what we try to do is facing a lot of difficulties and failures, but we should never probably give up and try to carry on and persevere. Obviously accepting that we have to change track sometimes, but still very important to keep that positive attitude.
Yeah, the key thing about making advances, if someone, one of my mentors once said to me that if you come up with an idea, a research hypothesis, an idea, a clinical approach, and at least one person doesn't tell you you're an idiot, you're probably not thinking enough. So it's when people say this is a stupid idea, this is blah, 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 that's the time you know that you're onto something. So what was the moment or experience that sparked your interest, Paolo, in hematology and the specific area you worked?
Well, when I think back, so I would say I was generally a very good student when I was in Italy. But when I started to work in the UK, because I moved straight after my graduation in the UK, I was mostly working on the clinical side. And there, when I first actually thought about doing hematology as a clinician, was one of the reasons was because I was doing an attachment as in an oncology department. Actually in a district general hospital. So they were actually doing both oncology and hemato-oncology. And there were consultants coming from the referral from the tertiary center into the district hospital to see patients. And I was helping out. And I liked always oncology as a student because of the biology around it. But I always kind of felt I don't want to do this because I'll see a lot of people suffering and I won't be able to make an impact. But within hematology, I actually noticed, and that's probably a bit unfair now to the oncology solid tumor field, because that's moved a lot. We're talking about back over 20 years ago almost now. But at the time, I felt in hematology, actually, you could make an impact. We could save some people with lymphoma already. We were doing very well, for example. And I felt, okay, I like this, because I'm still doing oncology, but actually, from a clinical point of view, I actually see the results of what we're doing.
Yeah.
And then afterwards, what I also started to think, mostly when I started to actually do hematology in the clinical side, was this is a perfect specialty to do research as well, because we have the tissue in our hands very easily. We don't have to do major surgery to get tissue studied. We just need the blood. We just can ask patients to give us some blood. Most of the times, depends on obviously on the type of cancer you study within hematology, and then you can get the tissue to do all to ask all the questions you want, and with the tools and technology, improving over the years, that's becoming more and more feasible. So I think those are the two main reasons possibly why I got fascinated by hematology to start with, and then to carry on as a physician scientist within hematology.
Sure, very much so. And your specialty allows, it's always struck me, the continuum from diagnosis to treatment. And hematology friends say, that what got them was they could take a blood sample, look under the microscope, and understand how understanding the biology, know that how that would change what they would see. So a random question here, Paolo. If you weren't a doctor, a physician, scientist, what might you be? What other career?
Again, difficult one. So there's two aspects I like about what I do. One is the scientific side, so the data analysis. So I could probably say something to do with that. But actually, the other aspect I like a lot is helping people. I still practice, despite doing a lot of research. I still practice, and I do like when that happens, which it doesn't happen all the time, but when it happens, that you make an impact on somebody's life, what you have actually proposed to do and what was done eventually as part of the team looking after the patient, that's resulted in those patients potentially being cured or having a good quality of life for as long as possible. And that is very rewarding. So I think possibly this is the aspect that still I like most of my job, and I find most rewarding still. So I would probably say probably something to do with helping somebody to develop, to improve. So maybe something like working in a school environment. I don't know. I mean, that I think that would be very rewarding. I know it's a tough job, but I see like school teachers for my kids, and I find what they do actually is really important. And if they do well, and when you come across a good teacher, that can make a huge impact in people's life. I mean, I remember some of my teachers, and I know other family members and friends who have come across teachers who have made a huge impact in their life.
You're right. Together with medicine, it's the one area that can have a profound impact on someone's life. So, tell us a common myth or a misconception about leukemia that you often find yourself correcting, whether that's with patients or with other doctors. I'm a surgeon, so what do I know? What misconception or myth might I have in my head that you could?
So there's two things, I think. One is with possibly junior doctor and medical students as well. The word leukemia obviously means white blood, as you know from Greek. And the reason why people always think leukemia, white blood, is because leukemia is you have very high white cell count, and as a result of that, then obviously when you look at the blood samples, the amount of red cells is reduced, and the blood is more white, if you want to say it like that. But the reality is that leukemia can present in a lot of different ways. And one of the misconceptions is that actually patients can present with acute leukemia with having actually low counts. And I often find that patients have a borderline low count, and that doesn't get investigated enough for a while until somebody picks up on it. And eventually you do a bone marrow, you find the patients have leukemia. And this is obviously a life-threatening condition. Obviously, when his low counts is probably less aggressive and acute than high count leukemia. But it's still something important that people are aware. So that's, I think, one misconception. And probably the other one I would say is that people often think of, as I said before, of these as being very aggressive, life-threatening, uncurable condition. But the reality is that actually we now can treat successfully, above all in young patients, I have to be honest, a good proportion, probably at least 50% of patients with what we have in terms of therapies. In, for example, acute myeloid leukemia, which is what I treat mostly, and that is something that I think needs to be communicated more and more, because people not need to be afraid of this as something that is necessarily a life sentence. It's a very serious condition. Don't get me wrong. It will shorten many people's life expectancy significantly, but we can do a lot of things to treat it, and I think that's important. People are aware of.
Yeah. I think back to my professional career and how our perception of this disease has changed. I've been involved in a kids' charity for many years, and I know that we used to see kids and they were getting their dying wish, basically, right? And now, okay, they've got a disease to deal with and it can become a chronic disease, but they're surviving. So thank you to you and your profession. So you've trained and worked in several prestigious institutions. What lesson or piece of advice has stuck with you throughout your career? I'll give you the one I was always told, which is put the patient at the center of everything you do, whether it's clinical work, research, whatever it might be, developing technologies, put the patient at the center. That's the one that's stuck in my brain. What about yours, Paolo?
I mean, I was thinking more, in different places I learned different things, and to work in different places, I think what I will say as advice is to try to learn from different places where you work what they are really good at. So if you work in a center which is like, I was lucky, for example, at some point to work at University of Cambridge in the hematology department, University of Cambridge, that is very biology of leukemia focus. It's probably not the biggest clinical center in the UK by far. I mean, the centers in London, which obviously treat many more patients that are stronger clinically. But in there, you really learn how to approach scientifically the problems of the biology of myeloid leukemias or hematological malignancies in general. And then I would say try to find what you are best suited for. There's no point in staying in a place just because of its prestige if you are not just a good fit for it. If you think you are better suited to work in a more clinical department, then don't stay in a very strong biological department. If you like a place where you have more interaction with patients, then go there than another place. So I think those are probably, I would say, but try to learn through your journey. It's probably good to move and see different things in order to actually as well learn all these things and decide where you want to go next and where you want to set up your own career eventually.
Yeah, interesting. So let's get into some more in-depth questions. Your research group investigates how adaptive metabolic changes influence both the progression of acute myeloid leukemia, AML, and resistance to therapy. Can you share what you've learned about these changes and how they differ from normal hematopoietic stem cells?
So yeah, thank you. So basically, the big advantages, the big developments in the field of leukemia over the last, I would say, almost 20 years now, has been the genomics, obviously, right? So we have learned a lot more about how the different leukemias are basically genomically defined, and that's to do mostly with technology. Obviously, that's the technology that's been first developed. And that has led to development of certain therapies targeting specific genomic alteration, above all if they are targetable. And that's helped as well to improve treatment outcomes in patients. But obviously what we learn is that genomic is not the whole thing. And often, beyond specifically hardwired genomic alteration, there's plastic changes that leukemia cells undergo, above all when stressed, for example, by a therapeutic intervention, to adapt to that and then to then become resistant to the therapy. And metabolism is specifically a very plastic type of process happens in every cell, which cells use to produce, energy, to produce building blocks for proliferations. And what we learned in the process has been effectively that leukemia cells, like many other cancer cells, to be fair, have changes in their metabolism, which sometimes are driven by genomic alteration, but sometimes are independent of genomic alteration. They are just common across many subtypes, genomic subtypes of leukemia, which can then allow them to withstand therapeutic stress. But they can become dependencies on their own, obviously, because then you can target these metabolic alterations to overcome therapy resistance. And we have done so already. If you think about it, that metabolic therapies have been used, for example, in leukemia. We use drugs such as asparaginase or methotrexate, which are metabolic therapies. So what is important, though, is, as you say, is what is the difference between the normal and the cancer? Because metabolism is common to every single cell in our body, even normal cells, obviously. And if we target it, we might get a lot of side effects. And we are seeing that, in fact. So I think a key point is to try to understand what is actually making the difference between normal and leukemia, and if something specific to leukemia which we can target. And that's what we try to do in the lab mostly, try to understand this and how to improve this targeting.
Okay, thank you. So, Paolo, you've got a background in studying both chronic myeloid leukemia and acute myeloid leukemia. What are the key biological and therapeutic distinctions between the diseases, and what's led you to focusing more recently on AML?
Very good question, actually. So they're both myeloid leukemia. So obviously there's some similarities, but the main difference is obviously that one, chronic myeloid leukemia, as the name as well says, is a less aggressive disease, but above all is a less complex disease. Chronic myeloid leukemia is a poster child for many things in AML, in hematology, sorry. And that includes the fact that it was one of the first leukemia where a specific genomic alteration was identified as being the driver of the disease, the famous Philadelphia chromosome due to the translocation between 8 and 21 chromosome leading to the BCR-ABL oncoprotein. And also the fact that this was then therapeutically targetable with small molecules. And we have used this now in CML for a long time. AML, although being myeloid as well, is often a much more complex disease. There's an average of 3 to 5 mutations in each AML sample. So it's genetically more complex, it's more heterogeneous. Within AML name, we have a lot of different subtypes, while in CML, everything is, reconciled by the presence of the BCR-ABL mutation. And as a result, obviously, AML is a much more difficult nut to crack, if you know what I mean. Like it's a more complex disease. So while CML is a fantastic model for many things, including developing of targeted therapies, also studying leukemia stem cell biology, because it's a very much model of leukemia stem cell type of malignancy. It's also possibly less of a clinical need, and that was probably one of the main reasons for me to move into AML, to study an area which I think was more an unmet need, and also to study something a bit more challenging in terms of therapeutic interventions. Because as I said, while I'm not saying that all CML is sorted, the vast majority of patients do well with current therapies. And I think in AML, instead, there's the scope to understand therapy resistance and develop better therapy more.
Right. More of an opportunity to make an impact. Yeah, that makes sense. So I want to drill down. There's you recently published a paper entitled, I'm going to go over the title of the paper, Steroyl-CoA desaturase inhibition is toxic to acute myeloid leukemia, displaying high levels of de novo fatty acid biosynthesis and desaturation. It's easy for me to say. Can you discuss this, please, Paolo, the implications of this?
Yeah, so this goes back to the previous question you asked about the interest we have in metabolism. And steroyl-CoA desaturase is a metabolic enzyme and is involved in, as the name says, desaturation of fatty acids. Now, the interesting things here are few. So first of all, normally in our body, there's few cells really that make fatty acid de novo, as we say. So they produce fatty acids from other nutrients like sugars or amino acids. And generally, these are the liver and the adipocytes. But for some reason, cancer cell, goes back to the concept I was telling you before, have a rewired metabolism, and they actually have quite an active fatty acid biosynthesis pathway. So this could be a difference between normal hematopoietic stem cells and leukemia cells, in that the leukemia cells have more active fatty acid biosynthesis machinery. And within that pathway, there's different proteins one can target. And we found that this desaturase, which is a specific step in the biosynthesis of fatty acids, which basically adds a double bond to some fatty acids, but it's very important to keep a balance between saturated and desaturated fatty acid, which is important for many reasons in a cell. So this enzyme was particularly important in AML. And what we learned was that it was prognostic in AML patients. It could be therapeutically targeted with clinical grade inhibitor, and we have used that in our manuscript, and this is now in phase 1 clinical trials. And not in hematological cancer yet, because it was developed for some soil cancer, but the plan is to move in hematological cancer as well. And basically this therapy seems to work, but only in a proportion of patients, samples that we've tested and models that we've tested, because this was preclinical work. And effectively, that goes back to the other point I was making before. Metabolism is important in many cells in our body. And I told you before, fatty acid biosynthesis happens in the liver as well. So maybe if you target this, you might have side effects in the liver. So I think we need to understand which AML really rely on this pathway and really become dependent on this pathway to possibly target this therapy so only to these people, because those are the people who will have a therapeutic window for us to be successful using this as a therapeutic target. And that's exactly what I think we learned from the paper, that only certain subtypes of AML were dependent. And now we are moving on this research further to try to understand if there are biomarkers actually which we can develop or use to leverage this therapeutic intervention. Because obviously it is enough in preclinical models in a lab to test fatty acid biosynthesis in AML cells, but doing it in a patient sample, a patient comes to the clinic, is more complex. So maybe we just need some markers which we can easily measure, which reflect how their AML is dependent on this pathway and whether that will be a good then patient to treat with this therapy.
Fascinating. You've published a number of papers on clonal hematopoiesis. Can you explain? I want to go to bed less stupid tonight.
So clonal hematopoiesis is a concept which was first described now just over 10 years ago in 2014. I remember still December 2014, when normally the big hematology meeting in the States happens. Two papers came out on the New England Journal at the same time, showing basically from big, collection of blood samples from healthy volunteers mostly, they could find in these patients by genomic sequencing mutations which are recurrently found in patients with myeloid leukemias. And basically, these people didn't have leukemia, though. They just carried the mutation. And what it transpired is that actually this mutation happened pretty much in each one of us, above all as we age, just as part of possibly aging process within our bone marrow. And they can sometimes just sit there for a long time without causing any problem. But in a small proportion of cases, they can progress to a myeloid leukemias. And what we've learned, effectively, this is a pre-leukemic condition. Effectively, these people are well. They don't have any problem with their blood. But if you carry this mutation, you are just slightly higher risk of developing leukemia. And all of us eventually will, if you look hard enough, will develop this as we age, as I said. But many of us will die for something else. But I think what this has prompted now is try to understand what happens in those patients that actually progress to leukemia, and can we do something to alter that progression? And so, for example, in some of the papers, there was one who just came out actually last week or 2 weeks ago, I can't remember now, in Nature, together with another two, where I'm not the leading author, to be fair, but I collaborated. But I found the findings were fascinating, and the three papers were giving the same message, that one of the most common mutation in clonal hematopoiesis called DNT3A becomes very reliant on metabolism, for example, and on oxidative phosphorylation from the mitochondria of these leukemia cells. And they actually showed, based on some UK biobank data, that was really, really fascinating, that patients who take metformin, which is known to inhibit the oxidative phosphorylation in the complex, the electron chain complex in the mitochondria, and is an anti-diabetic drug which we normally use and is very safe. These patients actually had the reduced incidence of clonal hematopoiesis driven by this specific mutation, prompting the idea that if you have a patient with this mutation who has a high risk of developing leukemia based on the level of this mutation in their blood, who still has clonal hematopoiesis, but we know that based on the level of the mutation, they might develop leukemia, maybe you can give them metformin to prevent this to happen. So as you see, now actually we're not just treating AML, we're moving towards actually trying to prevent it if we can, as we understand this process. So that's quite fascinating. I mean, it's early stages, and this is not been done clinical trials yet, although I'm sure it will happen very, very shortly. But it just goes to show the progress of the field in the last 20 years or so.
A lot of people working on metformin for a range of things, that it seems to be like a rather interesting drug. I've heard it described as vitamin M.
Yes, I think it's a bit like aspirin, right, or statins. They seem to be able to do. And now we have the GLP-1 agonist, the new kid on the block, which seems to cure everything.
Yes, again, fascinating drug. And I was just reading about the origins of it from the saliva from the Gila monster in the American Southwest. I love all this stuff. So let's get back to AML. As you've intimated, difficult disease to treat still, especially in relapsed or refractory cases. What are the biggest obstacles, do you think, to curing AML, or at least making progress in the treatment of AML? And how can we overcome those obstacles?
So obviously, I mean, what we work on is therapy resistance, and I mentioned that is a big obstacle, one that we don't fully understand yet, because we probably have focused mostly on genomic driver of this resistance, and mostly also as well on cell autonomous drivers of this resistance. So what I mean by that is that we specifically look at the leukemia cells and say how they adapt to therapy resistance. But often resistance can be helped or supported by the microenvironment, or sometimes, as we call it, by the macroenvironment. So, for example, the fact that if you have patients with changes in their, for example, metabolism at organismal level, or inflammatory processes, those can affect response to therapy. So we need to understand therapy resistance better. And the other thing we need to understand, I think, better in AML and in cancer in general, but I talk about AML because that's my field, I guess, is heterogeneity of the tumor. That's a big problem, because we often find that there's a lot of different mutations in AML. And we know from studies that they can happen in different clones. But when we treat patients, we don't really know that. We just see that mutations are present. We don't really know the clonal architecture of the cancer. And I think if we could improve that understanding in the clinical side and understand better the heterogeneity of the cancer, we can probably approach therapy slightly differently, because we will know that potentially certain therapies may help one clone, but actually select another one. And I think we need to find therapies which are either good across the board, or maybe combine them in a way that makes sense to tackle all the different clones. I think that heterogeneity is a big problem in cancer, obviously. And it's also plastic. It tends to change over time. So something to study and understand better, for sure.
Right, sure. So looking ahead, what excites you most about the future of leukemia research? Where do you hope to see the biggest, and where do you hope personally to make the biggest impact over the next 5 to 10 years of your career?
So as I said, I think understanding therapy resistance and developing better therapies, and that will require both identifying targets, but as I said, also identifying markers for patients likely to respond to certain therapies. So that getting better therapies is definitely something that I would like to develop. The other thing I would say is obviously understanding the heterogeneity better, and why patients with the same mutation sometimes can be having such different outcomes. So, you have, in our clinical practice, we see this all the time, but also in the studies, you can see that there's certain patients which on paper, they look the same in terms of their disease biology. They should be the same. And actually they do very differently. And why is that? And can we understand that a bit better? Because that would be very important in order to get the therapies right as well. So I think these are the things where I think we should focus mostly.
Yeah, a more personalized approach, and we see that in all cancers, don't we?
Yeah.
Variability. Well, Paolo, as we come to the end, finally, it's the final question I like to ask. It's a bit fanciful. If you came across a magical genie who gave you 3 wishes for your area of healthcare, what would the good doctor wish for?
Well, it's probably more than 3, but so I think I've said a lot of this, but I think we have made a lot of improvements in treatment of leukemia and AML in particular, being the disease I'm mostly focused on. But I think we probably, the big wish is to actually get even better to that and potentially get to a day where we can treat most patients, a bit like we do in CML these days, most patients with just a tablet, maybe a couple of tablets, and be able to give them a good quality of life for as long as possible. I mean, the other couple of ones, I'd say probably, it goes back to the first question you asked about Donald Thomas and how he did the research. And I know I'm not by any way, shape or form proposing we go back to that. I think research has to be ethical. But I think it's also to be said that I think these days trying to think and be able to deliver the things he did in our environment has become much more difficult. I think sometimes regulation and overburden of bureaucracy can actually take down a lot of good ideas and the willingness to actually try new things. And I think we should be practical sometimes, that there are situations where we really need to make big improvements. And in those situations, we probably need to accept that there's risk to be taken sometimes, because the outcome otherwise is going to stay that dire. And I think we need to make research and clinical research a bit easier. I think both preclinical and clinical is becoming very much burdened by a lot of administrative and bureaucracy burden. So I would like to see that reduced if possible, because I think that will lead then to the third point in my mind, which is very important. These are rare conditions, and we need bigger cooperation, and we need to be able to collect data from different countries and amalgamate those and learn from each other more. And sometimes this can be made difficult again by the administrative bureaucracy aspects. And I think those are things that we should hopefully improve, because all lead eventually to the first point of getting better outcomes, which is what I would wish for mostly.
So that's your wishes. You don't have any others. That's it.
Well, I mean, that's a personal ones, I guess, but that's all.
Well, your comment about collaborations, it's getting harder, and we would have hoped that it would be getting easier, of sharing data. I understand data privacy and scientific integrity, but it just seems like the people in power need to understand the implications. I like to say, we need to do things to help patients. And if everyone understood, we're all going to be patients sooner or later. So I just want to say thank you very much, Dr. Paolo Gallipoli, for spending your time talking to us, for everything you have done for patients and everything that you will continue to do, I am sure, for the science and practice of hematologic oncology. Thank you, sir.
Thank you. Thank you very much for having me here.
Well, I hope you enjoyed that episode of Hema Now, our bi-weekly podcast on hematology. Please consider subscribing on either Apple Podcasts or Spotify, or frankly, wherever you get your podcasts, so that you never miss an episode. Like us on social media, tell your friends and colleagues, and please check out the archives, because they're growing if you've missed any episodes. And please join us again in 2 weeks for another episode. Until then, I'm Dr. Jonathan Sackier. I'm proud to be your host. Please, everyone, stay safe, stay well, stay curious. Bye for now.